Hello friends, today we will gain knowledge about the topic detecting macromolecules of genetics, model organisms for genetic analysis, detection between phenotype and genotype, and our main objectives will be to study the macromolecules of different types, to gain knowledge of the detection of macromolecules of genetics, to study the model organisms for genetic analysis, and to understand the difference between phenotype and genotype. Friends, let me first give you a brief introduction about the term macromolecule. A macromolecule is a very large molecule made up of smaller units called monomers. The monomers may be the same or slightly different. Macromolecules are formed by dehydration reactions in which water molecules are removed from the formation of bonds. Four main types of macromolecules control all activities. They are proteins, carbohydrates, nucleic acids and lipids. Their monomers are amino acids, sugars, nucleotides and glycerol and fatty acids. Although the four categories of macromolecules contain different kinds of subunits, they are all assembled in the same fundamental manner. To form a covalent bond between two subunit molecules, an OH group is removed from one subunit and a hydrogen atom is removed from the other. For every subunit that is added to a macromolecule, one water molecule is removed. Energy is required to break the chemical bonds when water is extracted from the subunits. So, cells must supply energy to assemble macromolecules. Carbohydrates are important energy storage and structural compounds. The simplest sugars are called monosaccharides. They typically have 5 or 6 carbons arranged in a ring. An important 5-carbon sugar is the deoxyribose sugar that is part of the nucleotides that make up DNA molecules. Examples of 6-carbon sugars are glucose and fructose. Proteins are composed of carbon, hydrogen, oxygen, nitrogen and small amounts of other elements. They are polymers of amino acids which are joined together by peptide bonds. There are only 20 different amino acids that form all proteins. These specific amino acids include, and the order in which they are strung together determine the nature of a specific protein. This specific stringing together of amino acids is dictated by the genetic information encoded in DNA. Lipids are molecules composed predominantly of just carbon and hydrogen and are important structural and energy storage compounds in the bodies of living things. Unlike the other macromolecule classes and unlike water, lipids are nonpolar and therefore insoluble in water. Important lipid classes include fatty acids, phospholipids, steroids and waxes. Like other macromolecules, nucleic acids are polymers made up of monomers called nucleotides. Each nucleotide has 3 parts, a phosphate group, a 5-carbon sugar and a nitrogenous base. The phosphate group of one nucleotide is linked to the sugar of adjacent nucleotide in a phosphodiester bond. The nitrogenous base of each nucleotide in DNA can be one of 4 possible bases, that's adenine, thymine, cytosine or guanine. These 4 letters form the alphabet of the genetic code and remarkably can be code for almost an indefinite number of possible proteins and endless variety of biological forms. Nucleic acids fall into 2 general classes, deoxyribonucleic acid and ribonucleic acid. These molecules are responsible for all storage, expression and transmission of genetic information. In a simplistic sense, DNA tells how to make protein. Since the information encoded in DNA determines which proteins will be made and proteins determine all of the characteristics of an organism, you can think of the DNA as a set of instructions that tell exactly how to make a particular living thing. We will now study detection of macromolecules of genetics. Because the main macromolecules of genetics are DNA, RNA and proteins, genetic analysis often requires the detection of specific molecules of each of these three types. How can specific molecules be identified among the thousands of types in the cell? The most extensively used method for detecting specific macromolecules in a mixture is probing. This method makes use of the specificity of intermolecular binding in which a mixture of macromolecules is exposed to a probe that will bind only with the sought after macromolecule. The probe is labeled in some way either by a radioactive atom or by a fluorescent compound so that the site of binding can easily be detected. Now, let us look at probes for DNA, RNA and protein. Probing for a specific DNA. A clone gene can act as a probe for finding segments of DNA that have the same or a very similar sequence. For example, if a gene G from a fungus has been cloned, it might be of interest to determine whether plants have the same gene. The use of a clone gene as a probe takes us back to the principle of base complementarity. The probe works through the principle that in solution, the random motion of probe molecules enables them to find and bind to complementary sequences. The experiment must be done with separated DNA strands because then the bonding sites of the bases are unoccupied. DNA from the plant is extracted and cut with one of the many available types of restriction enzymes, which cut DNA at specific target sequences of 4 or more bases. The target sequences are at the same positions in all the plant cells used. So, the enzyme cuts the genome into defined populations of segments of specific sizes. The fragments can be fractioned by using electrophoresis. Electrophoresis fractionates a population of nucleic acid fragments on the basis of size. The cut mixture is placed in a small well in a gelatinous lab and the gel is placed in a powerful electrical field. The electricity causes the molecules to move through the gel at speeds inversely proportional to their size. After fractionation, the separated fragments are blotted onto a piece of porous membrane where they maintain the same relative positions. This procedure is called a Southern blot. After having been heated to separate the DNA strands and hold the DNA in position, the membrane is placed in a solution of the probe. The single-stranded probe will find and bind to its complementary DNA sequence. Southern blot method is named after its inventor, the British biologist Edwin Southern in 1975. Probing for a specific RNA. It is often necessary to determine whether a gene is being transcribed in some particular tissue. This determination can be accomplished by using a modification of the Southern analysis. Total mRNA is extracted from the tissue, fractionated electrophoretically and blotted onto a membrane. This procedure is called a Northern blot. The clone gene is used as a probe and its label will highlight the mRNA in question if it is present. The Northern blot is a technique used in molecular biology research to study gene expression by detection of RNA or isolated mRNA in a sample. With northern blotting, it is possible to observe cellular control over structure and function by determining the particular gene expression rates during differentiation and morphogenesis, as well as in abnormal or diseased conditions. Northern blotting involves the use of electrophoresis to separate RNA samples by size and detection with a hybridization probe complementary to part of or the entire target sequence. Northern blotting takes its name from its similarity to the first blotting technique, the Southern blot, named for biologist Edwin Southern. The major difference is that RNA rather than DNA is analyzed in the Northern blot. Probing for a specific protein. Probing for proteins is generally performed with antibodies because an antibody has a specific lock and key fit with its antigen. The protein mixture is electrophoresed and blotted onto a membrane. This procedure is called a Western blot. The position of a specific protein on the membrane is revealed by bathing the membrane in a solution of antibody obtained from a rabbit into which the protein has been injected. The position of the protein is revealed by the position of the label that the antibody carries. The Western blot is a widely used analytical technique used in molecular biology to detect specific proteins in a sample of tissue homogenate or extract. It uses gel electrophoresis to separate native proteins by 3D structure or denatured proteins by the length of the polypeptide. The proteins are then transferred to a membrane, typically nitrocellulose or PVDF membrane, where they are stained with antibodies specific to the target protein. The gel electrophoresis step is included in Western blot analysis to resolve the issue of the cross-reactivity of antibodies. Many reagent companies specialize in providing antibodies against tens of thousands of different proteins. Commercial antibodies can be expensive, although the unbound antibody can be reused between the experiments. This method is used in the fields of molecular biology, immunogenetics, and other molecular biology disciplines. Other related techniques include dot blot analysis, immunohistochemistry, and immunocytochemistry, where antibodies are used to detect proteins in tissues and cells by immunostaining and enzyme-linked immunosorbent assay. Friends, we will now study model organisms for genetic analysis. Model organisms are non-human species that are studied to understand specific biological phenomena with the expectation that investigations made in the organism model will provide insight in the workings of other organisms. Often, model organisms are chosen on the basis that they are amenable to experimental manipulations. Although many earlier approaches to biology emphasize taxonomy and the diversity of species, studies of specific biological systems have taken a central role in modern biology. Species that were easy to maintain and manipulate under artificial conditions were selected and came to be viewed as models for broad categories of taxa and sometimes for biological systems in general. Thus, a model organism represents its near relatives with respect to many attributes. It might even show facts or principles that are relevant to an entire domain of life. But how we define a model organism? Model organisms are often small, have a short generation time, are easy to work with in large numbers, and cheap to maintain, which facilitates experimental laboratory research and high throughput manipulation and screening. In most cases, Economics had an influence on the choice of an organism to study, such as agriculturally important species, for example rice, or those related to human health, for example the malarial parasite Plasmodium. All these species are receiving extraordinary attention from the research community and fall under the broad definition of model organism. Some examples of model organisms that have been widely used and their particular advantages include Escherichia coli. In the world of bacteria, the spotlight of molecular biology has fallen chiefly on just one species, that's E. coli. This small rod-shaped bacterial cell normally lives in the gut of humans and other vertebrates, but it can be grown easily in a simple nutrient broth in a culture bottle. E. coli copes well with variable chemical conditions in its environment and it reproduces rapidly. Its genetic instructions are contained in a single circular double-stranded molecule of DNA, approximately 4.6 million nucleotide pairs long, and it makes 4,300 different kinds of proteins. The fruit fly Drosophila melanogaster was first chosen as a model organism by genetist T. H. Morgan and intensively studied by generations of genetics after him. The fruit fly is small and easily grown in the laboratory. It has a generation time of only 2 weeks and produces many offspring. The nematode worm Caenorhabditis elegans normally lives in the soil but is easily grown in petri dishes. Only a millimeter long, it has a simple transparent body with only a few cell types and grows from zygote to mature adult in only 3.5 days. Its genome has been sequenced. It is easy to identify mutant animals. The frog Xenopus laevis serve as a major model for the study of embryonic development because the eggs are very large in size, develop externally from the mother and are able to withstand extensive surgical intervention for embryo manipulation studies. The zebrafish Denatured was more recently chosen and developed because it has a unique combination of advantages. It combines the advantage of large, accessible, easily observable eggs and embryos with advantages for carrying out genetic studies, that ease of breeding in the laboratory, and small simple genome. Arabidopsis thaliana. For studying the molecular genetics of plant development, an important model is a small weed Arabidopsis thaliana, small flowering plant related to cabbage and mustard family. Plant can grow and produce thousands of progeny after 8 to 10 weeks. Saccharomyces cerevisiae. Yeast are single-cell eukaryotic microorganisms instrumental to wine making, baking and brewing since ancient times. The function of many proteins important in human biology were first discovered by studying their counterparts in yeast. Mouse. Mouse has been used for many genetic studies because it is a mammal that is a relatively good model for human gene expression. The genetic and molecular organization of the mouse and human genes are quite similar. We will now study the detection between phenotype and genotype. A genotype refers to the actual set of genes that an organism carries inside. When these genes are expressed under observable conditions, they are called phenotypes and the expressions are called phenotypic expressions. Interestingly, isn't it, wondering how they are different? After all, how can a man be different from the genes he has inherited? The fact is, phenotypes are dependent on the genes they inherit. However, their expression is also influenced by the environmental factors. The influence of the environment modifies the role that the genes play to a certain extent. The expression of the genes modified by environmental factors produce a phenotype. So, genotype is an organism's full heredity information, while as phenotype is an organism's actual observed properties such as morphology, development or behavior. This distinction is fundamental in the study of inheritance of traits and their evolution. It is the organism's physical properties which directly determine its chances of reproductive output, while the inheritance of physical properties occurs only as a secondary consequence of the inheritance of genes. To properly understand the theory of evolution via the natural selection, one must understand the genotype-phenotype distinction. Similar genotypic changes may result in similar phenotypic alterations even across a wide range of species. An organism's genotype is a major influencing factor in the development of its phenotype, but it is not the only one. Even two organisms with identical genotypes normally differ in their phenotypes. One experiences this in everyday life with monozygous twins. Identical twins share the same genotype, since their genomes are identical, but they never have the same phenotype, although their phenotypes may be very similar. This is apparent in the fact that their mothers and close friends can always tell them apart, even though others might not be able to see the subtle differences. Further, identical twins can be distinguished by their fingerprints, which are never completely identical. The genotype can be determined through genotyping, that is the use of a biological assay to find out what genes are on each allele. The phenotype can be determined by observing the individual. Phenotypic plasticity. The concept of phenotypic plasticity defines the degree to which an organism's phenotype is determined by its genotype. A high level of plasticity means that the environmental factors have a strong influence on the particular phenotype that develops. If there is little plasticity, the phenotype of an organism can be reliably predicted from the knowledge of the genotype regardless of the environmental peculiarities during development. DNA phenotyping. It is the process of predicting an organism's phenotype using only genetic information collected from the genotyping or DNA sequencing. This term, also known as molecular photofitting, is primarily used to refer to the prediction of a person's physical appearance and biogeographic ancestry for forensic purposes. DNA phenotyping uses many of the same scientific methods as those being used for genetically informed personalized medicine, in which drug responsiveness and medical outcomes are predicted from a patient's genetic information. Significant genetic variants associated with a particular trait are discovered using a genome-wide association study approach, in which hundreds of thousands or millions of single nucleotide polymorphisms are tested for their association with each trait of interest. Predictive modeling is then used to build a mathematical model for making trait predictions about new subjects. This was all about today's lecture. Hope you have understood it well. Thank you.